Öncelikle herkese merhaba.
Good afternoon, everyone. Attention deficit hyperactivity disorders, genetics, and different types of the subtype characteristics will be discussed by this trial carried out in Turkey. I'm going to talk about the genetic differences. As you know, attention deficit is composed of two behavioral symptom clusters. One of them is lack of attention, distractibility, and the other one is hyperactivity and impulsivity. The total prevalence of this disorder throughout the world is 5.29%. However, although there are three different subtypes, clinically heterogeneity is observed in this neurodevelopmental disorder. Etiological heterogeneity is the reason for that, most probably, which means different pathways interact with one another in this pathogenesis. And in the last couple of years, many different disciplinary areas and trials carried out in those areas show that there are central nervous system abnormalities. However, genetics, environmental factors, neuronal processes, and their clinical reflections like phenotypical links between different components are not known well enough. Speaking from a genetic perspective about this disorder, we can say that the importance of this disease was understood before 1950 for the first time, if there is a psychiatric disorder in the family, you can even diagnose the situation better. So it is suggested that this is a hereditary problem, but the trials carried out later show that different environmental factors are effective, and disorders in many genes cause some predispositions. So the development and the advancing of the disease depends on the contributing factors, the number of those factors, and the interaction between those genes. There are two groups of trials. One of them is behavioral genetic trials. The other one is molecular genetic trials. Behavioral genetic trials are familial trials, twin trials, and adopted child trials. I think this is important in two senses. Firstly, as you know, familial disease is hereditary. Secondly, the child may be affected by the psychiatric diseases if parents or one of the parents have a disease. So the atmosphere within the family are important, and environmental conditions under which the child shall live gives us an idea about the structure of the family. If there is this diagnosis in one of the parents, the child has an 8 times higher risk of having disorder. In trials conducted in Turkey and abroad, we see the similar result. The twin trials on twins and adopted children are important mainly from a genetic perspective. The biological children are unfortunately acquiring higher risks, and the adopted children trials expectedly show that the incidence of this disorder among the parents is higher. The trials carried out on conducts show that the peer problem is mainly seen in monozygote twins. In 2010's meta-analysis, we see that the average monozygotes have a transfer rate of 71% and 41%. And this is one of the trials which has been replicated a lot. It is Faravan 2005. This average is 0.76%. This is the meta-analysis of 20 different trials. Of course, we see that the hereditary characteristics are higher than the genetic transfer of diseases like schizophrenia, and we see the importance of genetic disposition by those trials. When we look in terms of the subtypes and when we look at the twin trials, we see that the expected outcome is that if a subtype is really a different type. In homozygous twins, there has to be a concordance. Combined type and the inattentive type twins have different results, but the other siblings have combined type of probands. But for the type which has the combination, we cannot talk about the same thing. And hyperactivity impulsivity group, in half of those groups, we see that combined type is seen most of the time. And it supports a certain thesis. Hyperactivity and impulsivity is probably a primary subtype of this disorder, but maybe it is probably a different subtype, but this trial shows that it is a primary subtype. When you look at the molecular genetic trials, you see that there are two different molecular type studies. One of them is the linkage trial, the other one is the association trial. The linkage trials are usually trials where genes and chromosomes are evaluated. Association trials are carried out more in number, but the linkage trials are also carried out, and the most consistent trial results are found in 5p13 and 17p11, and predisposition in the disorder is increased because of these genes. These two gene areas are the most important. The association trials are increasing every day. They are mainly community-based, and segregation between generations are tested. Complex cases also show the same thing. Shorter genetic distances and a single gene is evaluated. Even the signal area of a single gene is being evaluated. Signal area polymorphisms, popovariant numbers can be evaluated. The candidate gene trials, the one that we see the most, can be seen on that table. All neuronal system related candidate gene trials have been carried out, but the most important ones are dopaminergic system trials. Dopaminergic system is the most important one, mainly because they have an action mechanism on drug treatment, and its pathogenesis and frontal limbic recovery like dysregulation, and many areas rich in dopamine are present. Therefore, dopaminergic system is the area on which there are many studies conducted. And the DRD4 gene is the gene which has been worked on the highest. The DRD4 receptor is encrypted. The frontal cortex and midbrain and amygdala are the places where we have the most of this one. Why DRD4? The most important reason, as I said before, is that the areas expressed have something to do, have a relationship with DEHP. In 1993, there was a trial carried out for novelties, and relationship between DRD4 were evaluated, and the authors have declared that high impulsivity and high level of arousability have a significant relationship with the level of DRD4. And it is another trial about migration patterns, and D4 gene shows that macro migrations and long distances enable the long alleles of D4 receptors higher in number. This finding is important for Turkey as well, because the HP incidence in Turkey is really high. Prevalence is higher when compared to the other communities. Maybe, as Sabri Ercan has mentioned, this is due to the fact that Turkish community is traditionally a nomad community. One of the genes has a name stemming up from the word Turkish. This might be an explanatory. When you write DRD4 gene, this is what you see in Google, these are small trials, not very large ones, but the titles are really sensational. DRD4 gene increases the risk of adultery, it says. Those who have DRD4 are more prone to cheating, according to those news. The adult DHB is seemingly problematic in terms of relationships. DRD4 genes were looked at in animals as well. Rats with DRD4 and without DRD4 were given 6-OHDA, and the rats who are given 6-DHDA with DRD4 have no behavioral control, but with the ones without the gene, there is a similar and standard withdrawal system. So DRD4 signal is necessary for the process to continue. Where exactly in DRD4 the studies were carried out? In exon 3 consecutive sequences were focused on. As you can see on this sequence, there are different repetitive repetitions between 2 and 11. The last one is 4 repetition. 4 repetition is what we see most frequently throughout the world, and then comes 2 and 7. This depends on the individual though. The receptor functions are also affected by those repetitions. When compared to 4 repetitions, 2 repetition alleles, dopaminergic receptor signal is lower than expected, according to this study. Neuropsychological and phenotype trials show that those genetic differences have an effect on the different types. Therefore, we can say that cognitive impulsiveness is mainly affected by this, but there are conflicting results as well, so we cannot say something definite. Imaging genetic studies, which are increasing in number these years, are giving us very important information. The sampling groups with this disorder and the control group composed of their healthy siblings show that prefrontal gray matter volume is affected in that group of patients. And at the same time, even in healthy controls, we see that there is a similar effect in siblings, although they do not have this disorder. Cortical thickness study is another important trial. The cortical thickness is affected by D4 receptors, receptor presence in DHB group, in DHD group, and in the control group. D4 receptor polymorphism is decreasing the thickness of the cortical area. How many trials are conducted in Turkey about the same issue? 4. Together with the one we've conducted, it makes 4. Tahir and his colleagues in 2001 have carried out a trial where they saw a significant relationship with 7 repetitions. This is a family-based trial. Families of the children with this disorder are included in this trial. 7 repetition polymorphism and DHD are found to be linked to one another, and this transfer relationship is found to be higher in those giving stimulus. What do we have in meta-analysis? OR is 95%. There is a significant relationship. However, small effect, an insignificant effect is what we see. So what do we see in our own trials? When we compare our trial with the other trials conducted in Turkey, we see that our sampling group has 3 subtypes: combined type, and the type determined according to DSM-5, and another group, the restrictive type. This is not in DSM-5 right now, but pure attention deficit group was what we saw in what we envisaged in that trial. We compared those with the control. For homozygote is higher in frequency in the second group, but in the restrictive group, 7 repetition allele is seen more frequently. Of course, those results are conflicting with one another. We cannot say something definite. 7 repetition can be dominant according to some publications, but some say that it has something to do with the combined type. Rowe and his friends say that the attention deficit is related to the dominant type, but Smith and his friends in their study have shown that DRD4 and the EHB subtype relationship shows that DRD4 7 repetition allele is more frequent in DEHB combined type cases. So in order to find those predispositions, there will not be enough of a potential in that patient group. In terms of 4 repetitions, there are different results and outcomes. Dopamine transporter gene is the other important gene. It is in the 5th chromosome, and it is responsible for the primary stimulation of dopamine. Why DHT1? The first reason is that one of the action mechanisms of drugs is that they inhibit the dopamine transfer. Secondly, the DHT1 carrying rats and rats which do not carry the same component are compared to one another. And there is an increased motor activity in the first group, and they respond to the stimulating agent treatment. In animal studies where the AT1 gene is non-functionalized, there is a decrease in dopamine in immunohistochemical staining. You see the tyrosine and hydroxin, which is reduced in number. Another study carried out on adults showed that the increase or decrease in dopamine transfer and the regression after methylphenidate is increased in the numbers on DHT1. We see that multiple repetitions are seen in certain areas, and these are the sites which are mainly focused on. Gene expression is expressed by those. The dopamine reuptake process has something to do with the dopaminergic pathway activity decreases. What do the meta-analysis say about DHT1? We see that the rate is very low. The positive relationship found in the first series of trials are losing their significance, and heterogeneity is also going down. How many trials do we have in Turkey about DHT1? One of them is we have 2 trials. One of them is from Sevinç's group, and the other one is Tahir's group. What do we see in our own trial? DHB and DHB subtypes are not related to DHT1 according to our trial, but in the literature, combined type is found to be related to the problem according to some studies. Why can't we find a relationship? DHB and image and impact trials show that in several sites, if there is a polymorphism, maybe DHT1 gene can be related. According to impact study, DHT1 has a moderator effect, therefore the effect can be seen during the progression of the disease. So this can be understood better in a longitudinal study. In comorbidities, Zoll and his colleagues in 2008 in their trial have shown that the study could be worked on in people with behavioral disorders. Now I have one last minute. I want to talk about another subject. This is the new study, a genome-wide association study is what I'm going to talk about now. This is the most popular study. Why are jivas so important? Because their effect is low, and DHB-like genes are causing many complex diseases, but they lack in terms of potential for the treatment of those diseases. The existing candidate gene trials can explain only 3 to 4% of the total phenotypic variance in ADHD. And simultaneously SNP and CNV are evaluated, and genes with the smallest effect can be defined. Human Genome Project HapMap project, CNV, individual genome analysis, 1,000 genome project are actually referred to as the revolution of the 21st century. Single nucleotide polymorphisms can be evaluated. Their penetration is very extensive, very low, excuse me, says the speaker. They do not cause any problem, but the mutation and penetration, mutations and penetrations are very rare, although they cause a significant disease. Related single nucleotide polymorphism related genes are seen on this table. Copy number variants are actually two equivalent DNA copies, one from the mother, one from the mother. After deletion and duplication processes, those DNAs can be more than expected or less than expected. DNA sites are newly discovered, and more than 3,000 copy number variants have been defined until now. And compared to single nucleotide polymorphism, they are more influential on the disease. And here you see the other trials carried out until now. In 2011, Topor and his colleagues have carried out another trial. Copy number of variants and single nucleotide polymorphisms are not overlapping on a single gene, according to those trials, but CHRNA7 gene or cholesterol or central nervous system development pathways and protein synthesizing genes are overlapping. Nicotinergic receptor alpha-7 is the most important one. For DHB, is this a candidate gene? We cannot answer that because in other neurodevelopmental problems we see the same thing. So while enlightening certain issues, this study is actually casting shadow on other issues. Is it enough to find a mutation? New trials show that it is not the case. More target-oriented and more brain-oriented studies have to be carried out, because in the animal trials we see that DHB's certain certain sites have something to do with learning, and the other sites have something to do with hyperactivity. If there is a dopamine receptor mutation in central complex, then hyperactivity can be seen. If in mushroom body we see the same mutation, we see learning deficits in those animal studies. So mutation exploration is not enough by itself. We have to look for the other mutations as well. This is the last slide, summary slide. This actually was the main article of our trial. 3 or 4 of the authors from the DSM-5 are listed here. I talked about the types and the subtypes of the ADHD, but when we look at the summary, we see that in terms of the subtypes of ADHD, the candidate gene trials show that attention deficit and hyperactivity are important in most of the cases, but there is no head-to-head comparison. And when one type is found, you may think that it is related to the other type as well. The clinical trials, clinical samplings are also important. ADHD and the type where the attention deficit is predominant, we still have lacking information. The point we've reached is just the beginning, and finding the genes severely will not be enough by itself. We have to think about the variations which are caused. We're at the beginning of this journey. We're going to find better results, because biological markers, when that happens, will lead us to new treatment modalities. Thank you.